So today's live lesson is about carbohydrates. We've been through a series on proteins, and today we're looking at carbohydrates. And carbohydrate series is going to get a bit shorter than the protein series. So it's just this week and next week. So today we're going to look at sort of basics and features and some ways to remember how to draw alpha and beta glucose. And then next week we'll have a look at linking structure to function. Okay, it's really obvious when we look at different polysaccharides, and it is such an important theme running through the whole of AS biology. So, right, for today, carbohydrates, the basics. So, let's call this one structure. We're working off CX H2O, Y. So this is then our general formula, and this is where we're talking about a chemical formula, not a structural formula. Okay, and all we're saying is that we're going to have a certain number of carbons, we're going to have a certain number of oxygens, and we're going to have double the number of oxygens to give us our hydrogens. So, for example, our classic example here is C6H12O6, which you come across in IG biology. This is glucose. And before that was it. Glucose was just glucose. Now you know glucose is a bit more than this. Okay, so before we get into glucose, let's cover a few more basics. We're talking about triose, pentose, and hexose sugars. Okay, so these ones have 3 carbons, these ones have 5 carbons, and these ones have 6 carbons. Now, if you think about a carbon chain, okay, you've got carbon, 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 and then you've got H's and OH's sticking out, right? Now, 3 is quite compact, but when you start adding these carbons onto here, the longer you get, the less stable you get. So, our pentose and hexose sugars have a problem. In chain form, they're unstable, less stable. So, we don't like instability because instability can contribute to lack of control, it can contribute to things going wrong. We want everything to be nice and stable and predictable. So this is why our pentose and our hexose form ring structures. It's all a very big deal about stability. Okay, so I'm going to actually then start another picture to talk about ring structures because I know I'm going to run out of space here. Right, so talking about now ring structures. Okay, so if we look at our pentose sugar, he ends up looking like this. Okay, so this is our pentose sugar, and how do you know I'm right? C1, C2, C3, C4, C5, 5 carbons. Okay, so I haven't drawn all the H's and all the OH's on here because I'm not looking at detail in those pieces. What I'm looking at here is just this ring structure. And you can see that that ring is a whole lot more stable than if it was just one strand. So a couple to be aware of here is fructose, because this is involved in sucrose, and sucrose is one of your big deals moving forward. Okay, and the other one, of course, is ribose, because he's a big deal when you get to DNA and RNA structure. So both fructose and ribose are pentose sugars, and we don't tend to go quite so much into learning the structure of these ones as we do into learning the structure of glucose. Okay, so thinking about glucose. First off, thinking about Our hexose sugars, here we have a slightly different structure because we have an extra carbon. Oopsie, there's a carbon and then another carbon on top. So again, we have a ring, we have our oxygen bridge. There we have an oxygen bridge here. So we're bridging with an oxygen to complete the ring. And we have one carbon sticking out the top. And we can do the same numbering exercise again. See, 1 is always this one on the right-hand side. 2, 3, 4, 5, and C6 pops up. And this is something that's important because when we talk about bonding with our carbons, we can talk about 1, 4 and 1, 6 and different types of bonding as identified by their carbon atom numbers. So it is important to have a good idea of where it's going. Again, I haven't drawn the H's and the OH's because at this stage we're just looking at this carbon structure. Plus, H's and OH's is where it gets fun for glucose. So this is our hexose, and our big deal here is glucose. Right, so I'm pretty sure that all of you have gone past this lesson already, so you are aware of alpha glucose and beta glucose. So let's just talk about them here as well. Alpha glucose and beta glucose. So these are two isomers of glucose, which means they have the same chemical formula, the same number of carbons, hydrogens, and oxygens, but they're arranged slightly differently. And as soon as you arrange something differently, you've changed its structure. And as soon as you've changed its structure, you change the way it interacts with other things. And as soon as you change the way the interactions happen, you change the function. Which is why it's so important that we link structure and function. And the twos can sort of help us learn and help us remember things because now we have a relevance for the structure. It's not just some random collection of letters. So let's draw our basic ring first, and then we can talk about how to remember which one of these is which. So both of them have exactly the same ring structure. Okay, and you think of this also in 3D. Okay, so you have your ring of 5 of your carbons and your oxygen, and then your CH2OH is sitting up. Okay, so CH2OH, he's above the ring. They don't make you draw this in 3D every time you draw it, which is lucky for you. Okay, right, now, alpha glucose and beta glucose, it's all about the hydroxyl group on carbon atom 1. Okay, that's where our change happens. Alpha and beta, A and B, and they're back to front. Okay, so alpha glucose or hydroxyl is below the ring. Beta glucose, your hydroxyl, is above the ring. So, how do you remember which way around they go? So, alpha OH below, beta OH above. So I've been sitting thinking, trying to get some sort of way of remembering this, and I came across something. Okay, what we want to do is go up, down, up, down, up, down. We've got a nice regular pattern. So if you remember that beta behaves, then if that one's up, this one is down, this one is up, and this one is down. And hey presto, we've got all our OHs in the right place. Alpha, of course, is identical because it's just that one hydroxyl group that's different. Okay, and then we can fill in our H's as well because we're being nice and thorough here. So opposite every OH, you have an H. Obviously, I can't talk and write at the same time. And now everybody has their correct number of bonds. So just now that we've finished, let us highlight the fact that OH is below the ring, OH is above the ring, and then here we've got OH, 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 OH. He behaves. He goes up, down, up, down. Okay, nice and regular pattern. So now you've got absolutely no excuse for not remembering which way around all your OHs and H's go. You have to be able to draw alpha and beta glucose. You have to be able to recognize them. You have to be able to pick them out of a lineup of molecules which you've never seen before. And you have to be awake because there's so much variety among molecules. Okay, we are not studying even 10% of what's going on out there. So you need to be thoroughly familiar with what it is you do study so that when it comes up, you can recognize it and you know what's going on and you know what you're doing with it. Okay, so we now have our structure of alpha glucose and our structure of beta glucose. And we've talked about rings and we've been nice and stable. So let us have a look now about what happens when we build these things together. So save that one. Clear everything. Okay, different color. Right, so we've talked about single rings. Okay, we've talked about glucose. We slightly touched on sucrose. Let's talk about this in a little bit more detail. So we have our monosaccharides, our disaccharides, and our polysaccharides. Okay, so mono is 1, di is 2, and poly is many, and saccharides are sugars. So 1 sugar, 2 sugars, many sugars. So are you a mono, di, or a poly when you have your cup of tea? Right, so few things on top of this. Our monosaccharides are our reducing sugars. Okay, our disaccharides are our non-reducing sugars. And our polysaccharides aren't even sugars. These are our more complex carbohydrates. Okay, storage and structural. So we're going to get more into that next week because we're going to look at what's the difference. Okay, what makes it a storage versus what makes it a structural? How is bonding all linked into that? Okay, so monosaccharides, reducing sugars, your classic example, glucose. Non-reducing, classic example, sucrose. So, seeing as I am running well with time today for a change, let's just digress here slightly and talk about testing for the biological molecules. Okay, so practical work is very much part of your AS biology syllabus. And yes, it is a case of following the instructions, but when it comes to biological molecule tests, they're not going to give you the instructions. They expect you to know those tests. So let us have a little bit of a recap of what's going on with reducing sugars test and a non-reducing sugars test. So if you are testing for a reducing sugar, you are going to add Benedict's solution, Benedict's reagent. You are going to heat. Now at IG, your teacher would have been quite happy if you just said, put in a hot water bath. At AS, that's not going to get you anything, okay? You have to have Benedict reagent and you need heat. You need a boiling water bath. And what you're going to get your color change. So we start with blue. We go through some sort of greeny, yucky, muddy colors. Yellowish, we get orange, and we get, at the end, brick red. Now, here's the catch. Again, brick red precipitate indicates the presence of reducing sugars. That's your classical basic phrase. But if you're testing for reducing sugars and your solution go green, does it have reducing sugars? Yes, it does. Okay, what it has is a little bit of reducing sugars. So these colors indicate increasing concentrations of reducing sugars. So if I'm starting out, I'm going to make a really concentrated, lovely glucose solution, and I'll give it to my students, and it's going to go poof, red to brick, blue to brick red, and everybody's going to be happy because they see it. Okay? If I want to be more subtle, or if I'm looking at different concentrations on purpose, then they're not all going to go brick red. They're going to go different colors, and they're going to do that at different speeds. Okay, so you got to bear in mind final color and speed of color change. And when your battery indicator changes to red, you also must plug in your laptop. Right, so there's quite a bit more detail, and if you go into the fact lessons about biological molecules, it also tells you more about how you can look at the time to the first color change to compare different concentrations. And remember, that first color change is not brick red. That first color change is this first greeny, muddy, yucky color. It's as soon as we're not crystal clear blue anymore, then we've got to precipitate. Okay, so disaccharides, a non-reducing sugars test. Well, reducing sugars test, but with a few extra steps first. Because remember, our disaccharide, let's, for example, use sucrose. So here's looking something like this. Okay, remember we have a hexose and a pentose stuck together. Fructose plus glucose gives us sucrose. And this is not an explicit thing that you need to learn, but it is something that you're going to come up against again and again. So it's well worth knowing, because it just makes it more familiar and easier when it does come up in the practical. So, monosaccharides, one ring. Okay, they can react with our Benedict's reagent and they can reduce it. Okay, here with sucrose, first thing we've got to do is we've got to get it into monosaccharides. So that is why we do the heat gently with hydrochloric acid. Okay, and what that does is basic chemistry. We're breaking bonds. Okay. Maltose is also one of those ones that comes up. Sorry, there's a question come up through about maltose. It comes up more in theory, doesn't come up so often in the pracs, but it would link into digestion of starch. So we can be playing with maltose if we're digesting a polysaccharide all the way down. Okay, we're going through maltose, and it's all going to work roughly in the same way. Okay, so something that you're bearing in mind here again is that you're learning this at AS level. You're still not learning everything. Unfortunately, as much detail as we learn it in now, it's still simplified from the whole world out there. Okay, so sucrose, we have to heat it gently with hydrochloric acid. We have to break that bond in order to get our glucose out so that we can then do our Benedict's test. But Benedict's is not a fan of an acidic environment. So after you heat gently, you're going to have to neutralize the acid before you carry on with your Benedict's test. Okay, so then gonna Benedict's as for reducing sugar. Okay, so I say don't worry so much about maltose at this stage. Next thing you know, you're sitting in your exam and there's maltose. Okay, and then you're thinking, she told me not to worry. And that is one of the classic examples of, remember that they're not necessarily only throwing things at you that you are familiar with. Okay, so you are familiar with glucose, you are familiar with sucrose. Now they're going to throw maltose at you and you see what happens. Okay, so it's really important that you follow the instructions in your prac. First off, don't make assumptions about things that you haven't worked with before. So if you're given maltose, don't assume it's going to act like this or it's going to act like that. Okay? Follow the steps in the practical and interpret your results. Okay? If you can follow instructions, if you're not very good at following instructions, start baking. Okay? Especially really picky, difficult cake recipes, because that will get you a lot better and a lot of practice about following instructions. Okay, it's exactly the same concept when you're in a prac. To work according to the instructions they've given you, they're not going to throw you in the deep end with no idea of what to do. They are going to guide you on what you should be doing. And it's up to you to read carefully and follow those instructions and pick up on the signals that they're sending. Okay, so polysaccharides, you've got a wild guess there that you've got a same test, but we've got to break it up first. So we can digest our starch, turn it into maltose, turn it into glucose, take it all the way down, and so on and so forth. Benedict's reagent. Okay, Benedict's just requires a reducing sugar. So, 7 minutes left. Let us talk about the basics of bonding within our carbohydrates. A different color again. Okay. Right, so bonding. You can even spell bonding, it helps. Okay, 3 important words. And glycosidic bond is your carbohydrates only, but these other 2, condensation and hydrolysis, are going to come up again and again and again in everything you're going to study. Okay, so condensation is loss of H2O. Hydrolysis is addition of that same molecule, H2O. Okay, however we are moving, we are controlled by enzymes. Nothing ever happens randomly. We want to build a starch molecule, so we're building amylose and amylopectin, we're adding glucose onto there. That reaction of adding the glucose onto the terminal end is enzyme controlled. If we want to release the glucose so that we have him available for respiration, that release of that glucose is enzyme controlled. Every time you form a disaccharide, enzyme controlled. Every time you break a disaccharide, enzyme controlled. Except, think about it, what did we just do? We broke sucrose with hydrochloric acid. So there we used chemistry and we used acid rather than using enzymes. But same effect, okay? We had to heat that as well to make it happen a little bit quicker. Enzymes don't have to heat it. Brilliant stuff. As long as we're at the optimum temperature, we're flying. Right, so we digress. Bonding, condensation, hydrolysis, addition of water, loss of water. The glycosidic bond is an oxygen bridge. And let us do it alpha glucose for now. So carbon, oxygen, carbon, 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 CH2OH. Okay, then we have another carbon, carbon, oxygen. Okay, something just flashed up on my screen. Okay, it flashed up behind some other stuff, but it was some sort of Zoom alert. So, just so that everybody's aware, if you've got questions, pop them into the chat, because that's the only way you can communicate with me during a live lesson. Okay, So we're working with alpha glucose, so we are below the ring. And beta is the one that behaves. So beta is above, so then we go below, above, below. I like this new way of remembering this. It does actually work. Right, and we fill in our hydrogens because we're very good students and we're very thorough. There, and one more. Okie dokes. So the question that was coming through is condensation breaking of bonds and hydrolysis formation of bonds. Not quite, it's the other way around. Okay, so condensation forms a bond. Hydrolysis breaks the bond. Okay, so what we're going to do now is a condensation reaction. We've got our two monosaccharides, we're going to build a disaccharide. So let us get another color for fun. Okay, so we want to remove an H2O. See how these two OHs are lined up so nice and friendly next to each other. Wonderful thing about alpha glucose. And look there. Okay, I'm going to change this slightly because I want him to sit nicely. Oh gosh, you know, one day I'm going to get used to this stupid eraser on this thing. Okay, so OH, because remember the O is what links to the carbon. Now we've got it drawn pretty. Now we can circle nicely, and our circle has two H's and one O. So this is H2O lost, removed, which is condensation. Now, the problem is when we lose that H2O, this carbon needs something to bond to. This oxygen needs something to bond to because carbon must bond to 4, oxygen must bond to 2. So now we have to, instead of being lost, they draw to each other. He bonds to the oxygen and then the oxygen is still joined to his other carbon. Now, I have really managed my space very badly. So let's just do our ring structure at least. I'm losing track. Goodness, somebody should have shouted quicker than that. My ring comes off my carbon. There we go. And this one also comes off the carbon, not off the oxygen bridge. There's the other oxygen, and there's the other carbon. So our glycosidic bond is this here, where we have removed our water. And it's been lost. So we need one more arrow that goes down to those. So you would draw all your OHs and Hs in on your second lot as well, but that is bad base management on my side, so they don't fit. But yeah, that shows you where that water comes from between those two molecules and how then that carbon and the oxygen have no option other than to join together so that they still have their correct number of covalent bonds to keep all of their electrons and orbitals and all of that chemistry nonsense happy. Okay, so somehow we managed to get it all in time. If you do have any questions else related to the lesson, then please feel free to send through a question on the chat and we can continue it further or in the Q&A tomorrow. If you want a copy of the notes, please remember to send me a message. Otherwise, I won't remember to send them to you. So, other than that, have a wonderful Tuesday. I might see you just now for maths. Otherwise, I'll see you tomorrow for bio or chat to you on the chat. Have a great day, guys. Bye.